The idea that gut bacteria directly influence energy and aging might seem surprising at first, but scientific research continues to reinforce the reality that our microbial communities are integral to our physiological function. These trillions of microorganisms, primarily housed in the gut, are not passive passengers. They are active participants in nearly every system that governs health, especially when it comes to how we generate energy, combat cellular decay, and ultimately determine the rate at which we age. Within the microbiome, certain bacterial strains stand out for their influence on longevity and energy regulation. Firmicutes and Bacteroidetes are two of the dominant bacterial phyla that play critical roles in breaking down complex carbohydrates, producing essential vitamins, and modulating immune responses. More recently, bacteria like Akkermansia muciniphila have gained attention for their ability to strengthen the gut lining, regulate metabolic efficiency, and improve insulin sensitivity, all of which are tightly connected to anti-aging mechanisms. When these beneficial bacteria are present in optimal ratios, they produce short-chain fatty acids like butyrate, acetate, and propionate. Butyrate in particular has demonstrated remarkable potential in reducing systemic inflammation, promoting cellular repair, and enhancing mitochondrial function, the latter being essential for sustained energy and biological vitality. Mitochondria, often described as the powerhouses of our cells, are responsible for generating ATP, the currency of cellular energy. However, mitochondria are highly sensitive to their environment. When inflammation is high or when oxidative stress is unregulated, mitochondrial efficiency drops and energy output declines. This is not only experienced as fatigue, but also manifests biologically as accelerated aging. Beneficial gut bacteria help prevent this scenario by creating a biochemical environment that supports mitochondrial health. They regulate oxidative stress, produce critical cofactors, and even influence the expression of genes involved in mitochondrial biogenesis. On the flip side, when the gut microbiome becomes imbalanced, a state known as dysbiosis, there is often an overgrowth of pro-inflammatory bacteria. These unwanted microbes produce endotoxins like lipopolysaccharides, LPS, which can leak into the bloodstream and trigger chronic low-grade inflammation. This inflammatory cascade doesn't just affect the gut, it reaches the brain, heart, muscles, and virtually every organ system. Over time, this internal stress burden wears down the body's energy systems, accelerates telomere shortening, increases cellular senescence, and hastens aging. The connection between gut bacteria and aging becomes even more compelling when looking at how microbial diversity correlates with lifespan. Studies of centenarians, people who live past 100, often reveal a microbiome rich in diversity and abundant in certain anti-inflammatory species. This microbial signature is associated with lower incidences of metabolic disease, cognitive decline, and immune dysfunction. In contrast, aging populations with depleted microbial diversity show increased markers of frailty, cognitive loss, and systemic inflammation. Another powerful element of this microbial relationship is the communication between gut bacteria and the brain, commonly referred to as the gut-brain axis. The microbes in our intestines send signals through the vagus nerve, the immune system, and hormone-like substances known as cytokines and neurotransmitters. For example, certain bacteria can produce GABA, serotonin, and dopamine precursors, all of which directly influence mood, stress resilience, and motivation. These mental and emotional states are not separate from energy. They are core components of it. A person's perception of vitality or fatigue is deeply intertwined with these neurochemical signals, which are regulated in part by gut flora. Ultimately, understanding energy and longevity through the lens of the gut microbiome offers a paradigm shift. It reveals that our health is not just genetically predetermined or based on isolated organ systems, but rather orchestrated by a highly adaptive network of microbial life that responds in real time to how we eat, sleep, move, and breathe. Supporting the optimal balance of these bacteria is not only about digestion, it is about fundamentally aligning with the body's natural blueprint for sustained energy and extended life. Short-chain fatty acids, S. esquifaes, particularly butyrate, are among the most powerful biological byproducts produced by beneficial gut bacteria. And their influence on human health, energy, and longevity cannot be overstated. These compounds are not ingested directly. They are created when certain bacteria ferment dietary fibers and resistant starches in the colon. Once produced, sequences like butyrate, acetate, and propionate act as signaling molecules and metabolic fuels, deeply influencing cellular function, inflammation, brain health, and the aging process. Butyrate stands out as the most impactful SCFA in terms of anti-aging energy regulation. One of its primary roles is to serve as the preferred energy source for colonocytes, the cells that line the colon. When colonocytes are fueled by butyrate, they maintain a healthy gut barrier, preventing the leakage of harmful substances like lipopolysaccharides, LPSD, into the bloodstream. This tight gut barrier is essential because when LPS enters systemic circulation, it triggers chronic low-grade inflammation, a driver of metabolic dysfunction, fatigue, and accelerated aging. Butyrate essentially fortifies the gut from within, helping to reduce systemic inflammatory load and maintain metabolic integrity. Beyond the gut, butyrate has a profound effect on mitochondrial function. Mitochondria are the cellular engines that produce ATP, and their health is essential for maintaining energy levels, cellular repair, and resistance to oxidative stress. Butyrate enhances mitochondrial efficiency by promoting the expression of genes involved in mitochondrial biogenesis and by supporting oxidative phosphorylation, the most efficient process for generating ATP. This means that butyrate doesn't just support gut health, it supports systemic energy production at the most foundational level. When mitochondria operate efficiently, energy is abundant, mental clarity is improved, and the wear and tear associated with aging is reduced. Another critical function of butyrate is its ability to inhibit histone deacetylases, HDACs. HDACs are enzymes that regulate gene expression by altering the way DNA is packaged within the cell nucleus. When butyrate inhibits HDACs, it promotes the expression of genes associated with antioxidant defense, immune regulation, and cellular longevity. This epigenetic influence is a major reason why butyrate is increasingly being studied as a compound with anti-cancer, neuroprotective, and anti-inflammatory effects. Essentially, it tells the body's cells to turn on the genes that keep them young and resilient while silencing those that lead to degeneration. SCFAs also play a pivotal role in metabolic regulation. Propionate and acetate, for example, are involved in gluconeogenesis and cholesterol metabolism. They help the liver regulate blood sugar levels and maintain healthy lipid profiles, two key aspects of preventing age-related diseases like type 2 diabetes and atherosclerosis. Butyrate, on the other hand, improves insulin sensitivity and has been shown in animal studies to prevent diet-induced obesity. These effects are not isolated. They are part of a systemic rebalancing driven by microbial metabolites that communicate directly with host tissues. Moreover, SCFAs influence the central nervous system. Butyrate can cross the blood-brain barrier and has been shown to reduce neuroinflammation and support the production of brain-derived neurotrophic factor, BDNF, a molecule essential for neuroplasticity, learning, and memory. This link between gut-derived compounds and brain health is a cornerstone of what's known as the gut-brain axis. When butyrate levels are high, cognitive function tends to improve, mood becomes more stable, and resilience to stress increases, all of which contribute to the subjective and biological experience of energy and vitality. The production of SCFAs, however, is contingent upon diet and lifestyle. Dietitian fiber from a diverse range of plants, nuts, seeds, and legumes provide the raw material for these compounds. When fiber intake is low, SCFA production plummets, gut health deteriorates, and the systemic benefits begin to unravel. This highlights a simple but profound truth on the way we eat doesn't just affect digestion, it influences cellular energy, gene expression, and the pace at which we age. And at the center of it all is butyrate, quietly orchestrating a symphony of biochemical signals that keep the body running at its best. The microbiome is an incredibly dynamic ecosystem that responds rapidly to environmental cues, especially those related to our daily behaviors. What this means is that the choices we make, what we eat, how long we sleep, when we get sunlight, how often we move, and how we manage stress are not just lifestyle decisions, they are microbial programming tools. Every behavior communicates something to the trillions of bacteria living within us, and in turn, those bacteria shape the state of our immune system, metabolic efficiency, cognitive clarity, and overall lifespan. The concept of aging is therefore not linear or inevitable. It is a reflection of how well we align with the biological systems that determine cellular maintenance and energy production, systems that are tightly regulated by the microbiome. The foods we consume, for instance, are not merely processed by our digestive enzymes, but are largely fermented, digested, or transformed by gut bacteria. A fiber-rich diet, especially one high in diverse plant-based fibers, leads to a more diverse microbial population. This diversity is one of the most consistent markers of a healthy, long-living individual. Diverse bacteria produce a broader range of short-chain fatty acids, vitamins, and neurotransmitter precursors. They protect the gut lining, regulate systemic inflammation, and even help modulate hormonal balance. In contrast, diets high in processed foods, sugars, and artificial additives tend to reduce microbial diversity and encourage the proliferation of pathogenic bacteria that trigger inflammation and cellular aging. Sleep is another major player. Studies have shown that even a single night of poor sleep can disrupt microbial balance, increasing inflammatory markers and reducing insulin sensitivity. Consistent, high-quality sleep, on the other hand, allows for microbial cycling that mirrors our circadian rhythm. Beneficial bacteria flourish during specific phases of the sleep-wake cycle, particularly during deep sleep when the gut lining repairs itself. This cycle of microbial renewal reinforces the integrity of the gut barrier and helps reset hormonal systems that govern hunger, satiety, and energy metabolism. When sleep patterns are irregular, this repair process is disrupted, leading to a cascade of issues like brain fog, mood swings, and accelerated aging. Sunlight also plays a surprisingly powerful role. Exposure to natural light, particularly in the early morning, helps regulate the body's circadian clock through the retina and brain pathways. But this same exposure influences microbial function indirectly by modulating cortisol and melatonin rhythms. When cortisol spikes in the morning and melatonin rises in the evening in response to proper light exposure, the gut environment becomes more stable, predictable, and conducive to microbial health. Moreover, vitamin D synthesis, triggered by sunlight, has direct antimicrobial properties and supports immune modulation, both of which influence the microbiome's ability to ward off pathogenic invaders. Movement, or lack of it, also alters microbial expression. Regular physical activity increases microbial diversity and boosts the abundance of specific strains that produce anti-inflammatory compounds. Exercise improves gut motility, reduces visceral fat, and creates a more oxygen-rich environment in the colon, all of which are favorable conditions for beneficial bacteria. Sedentary behavior, by contrast, slows down digestive processes, fosters stagnation, and allows harmful bacteria to flourish. This not only affects digestion, but can lead to low-grade inflammation and reduced metabolic efficiency. Stress management is perhaps the most underestimated microbial modulator. Chronic psychological stress increases cortisol levels, which in turn disrupts gut permeability, allowing unwanted compounds to leak into the bloodstream, a phenomenon often referred to as leaky gut. Stress also decreases the production of secretory immunoglobulin A SIGA, an antibody that helps regulate microbial populations. Practices like deep breathing, meditation, cold exposure, and even social connection have been shown to positively influence microbial balance by reducing cortisol and supporting parasympathetic nervous system dominance. Altogether, these behaviors form a feedback loop between the host and its microbiota. When we align our habits with natural biological rhythms, eating whole foods, sleeping well, getting natural light, moving our bodies, and managing stress, the microbiome responds by producing compounds and signals that enhance vitality, slow cellular aging, and preserve mental and physical energy. The microbiome is not just influenced by us, it is a reflection of us. And in many ways, our future health is being shaped day by day by the microbial ecosystem we choose to support. Intermittent fasting is often praised for its effects on weight loss, metabolic health, and insulin sensitivity, but one of its most profound yet less widely understood benefits lies in its impact on gut health and microbial balance. Strategic fasting, specifically time-restricted feeding or extended overnight fasts, creates a unique internal environment that not only enhances human physiology, but also significantly benefits the populations of beneficial bacteria in the gut. This microbial support translates into better digestion, reduced inflammation, stronger immune function, and, crucially, aging effects at the cellular level. When we fast, We initiate a temporary state of metabolic stress that encourages the body to shift from glucose-based energy production to fat-derived energy. This switch activates a cascade of beneficial processes such as autophagy, reduced insulin levels, and improved mitochondrial function. At the same time, fasting leads to changes in the pH nutrient availability within the digestive tract, which directly affects which bacteria can survive and thrive. Many pathogenic or inflammatory bacterial species rely on a constant intake of simple carbohydrates and sugars. When those inputs are reduced or removed through fasting, the population of these harmful microbes declines. In contrast, beneficial bacteria such as Akkermansia mucinifila and certain Bacteroidetes strains thrive under these fasting conditions, promoting gut barrier integrity and reducing systemic inflammation. Akkermansia mucinifila is particularly notable. This bacterium feeds on the mucus lining of the gut, not in a harmful way, but in a way that encourages the body to regenerate and strengthen that protective barrier. Fasting enhances this cycle by creating a period of low external nutrient input, which encourages microbes like Akkermansia to do their job more efficiently. A stronger gut lining means fewer toxins leaking into the bloodstream, less immune overactivation. Key benefit of intermittent fasting is that it encourages microbial cycling, vital for slowing just as we benefit from sleep-wake rhythms. Our microbiome also benefits from periods of feeding and rest. Constant eating, especially grazing across 16 or more hours a day, disrupts these microbial rhythms and can lead to dysbiosis, a condition where harmful microbes dominate and beneficial ones decline. Intermittent fasting reintroduces a natural rhythm, allowing the microbiome to reset, rebalance, and focus on repair during fasting periods. This rhythm also enhances the production of short-chain fatty acids during feeding windows, which are critical for energy regulation and inflammation control. Fasting also appears to upregulate genes associated with longevity, not only in human cells, but in microbial populations as well. When the digestive system gets a break, both the host and the microbes initiate protective processes. These include increased expression of sirtuins, FOX proteins, and other pathways associated with DNA repair, cellular resilience, and mitochondrial renewal. In animal studies, intermittent fasting has led to a measurable increase in lifespan, and while human research is ongoing, early findings suggest similar benefits, largely due to this intricate host-microbe interaction. Moreover, the benefits of fasting are not limited to the digestive tract. The gut-brain axis is also heavily influenced. During fasting, the microbiome shifts toward producing higher levels of GABA and serotonin precursors, which stabilize mood and reduce anxiety. Cognitive clarity often improves not just from ketone production, but from the neurochemical influence of a more balanced gut microbiome. This mental clarity feeds into energy perception. People feel more focused, more stable, and more motivated during fasting states, especially once their microbiome adapts to the routine. It's also important to understand that fasting doesn't require extreme duration to be effective. Even daily fasting windows of 12 to 16 hours can yield measurable benefits. What matters is consistency in allowing the body and microbiome to adapt over time. Gradually incorporating time-restricted feeding, such as stopping food intake after sunset or delaying breakfast by a few hours, can help promote microbial diversity, improve gut health, and slow down the processes associated with cellular aging. When used thoughtfully, intermittent fasting becomes less about restriction and more about rhythm, one that deeply benefits not just metabolism, but the entire biological system that sustains youth and energy. Cold exposure, light cycles, and circadian alignment are often seen as external variables, but they have profound internal consequences, especially for the microbiome. These environmental cues are not just passive influences, they actively shape the microbial populations in our bodies, and by extension, impact everything from immune regulation and inflammation to metabolism, hormonal balance, and aging. The microbiome responds dynamically to cues from our surroundings, and when those cues are consistent and supportive, like regular light exposure and intentional cold exposure, they create a biological environment that fosters resilience and longevity. Cold exposure, in particular, influences microbial activity through a few critical mechanisms. When the body is exposed to cold, even briefly, it activates brown adipose tissue and back, a type of fat that generates heat by burning energy. This activation is not just about thermoregulation. It has downstream effects on metabolism and inflammatory signaling. Studies have shown that cold exposure can increase the relative abundance of beneficial bacterial strains that support metabolic health and reduce systemic inflammation. These microbial changes lead to improved insulin sensitivity, enhanced mitochondrial function, and better energy utilization. Additionally, cold exposure appears to influence gut permeability and gut-brain communication by altering the microbial production of neurotransmitters and anti-inflammatory compounds. Light exposure plays an equally important role. Our bodies are tightly synchronized with the circadian rhythm, a roughly 24-hour cycle that governs hormonal release, metabolism, and cellular repair. The gut microbiome also follows this rhythm, shifting its activity levels based on the time of day. Early morning exposure to natural light is a critical synchronizer for the circadian clock. When light hits the retina, it sends signals to the suprachiasmatic nucleus (SCN) in the brain, which in turn regulates the release of hormones like cortisol and melatonin. These hormonal shifts influence the gut microbiome indirectly. For example, melatonin isn't just a sleep hormone, it also has antimicrobial properties and modulates gut motility. Consistent light, dark cycles help beneficial bacteria follow predictable patterns, allowing for optimal nutrient processing, detoxification, and repair during rest periods. Disruption of this light-based rhythm, such as from shift work, late night screen exposure, or irregular sleep schedules, can throw the microbiome into disarray. This disruption has been linked to increased levels of inflammatory microbes, reduced microbial diversity, and heightened risk of metabolic disorders. Restoring the circadian alignment through consistent light exposure during the day and darkness at night helps reverse these effects. Even seemingly small actions like getting sunlight within 30 to 60 minutes of waking can anchor the body's internal clock and optimize microbial function throughout the day. Circadian rhythm isn't just about light and dark. It also depends on the timing of behaviors like eating, sleeping, and moving. The microbiome responds to these behaviors by shifting its composition and activity. For example, eating late at night or having irregular meal times can desynchronize the microbiome's natural rhythm, leading to digestive inefficiency and increased gut permeability. On the other hand, aligning meal times with daylight hours and maintaining a regular sleep schedule reinforces a microbial environment that supports longevity. The consistency of these patterns signals safety and predictability to the body, allowing it to allocate energy to repair and regeneration rather than stress response. One fascinating aspect of this alignment is how it influences the production of microbial metabolites. When the circadian rhythm is stable, the microbiome produces more short-chain fatty acids like butyrate at the right times, especially during sleep when repair processes are most active. These metabolites support mitochondrial health, reduce inflammation, and even regulate gene expression linked to aging. But when circadian rhythm is disrupted, this production becomes erratic or suppressed, compromising the body's ability to maintain cellular and microbial health. Ultimately, cold exposure, light cycles, and circadian alignment are not just wellness trends or peripheral habits. They are deeply embedded in the body's biological design. They shape the microbial environment in ways that directly affect how we feel, how much energy we have, and how slowly or rapidly we age. By intentionally engaging with these environmental levers, we gain influence over internal systems that were once thought to be purely genetic or automatic. The microbiome responds rapidly and powerfully to these signals, and when those signals are aligned with our evolutionary biology, the result is enhanced vitality and extended health span. The future of anti-aging is unfolding not in corporate laboratories, but within the living ecosystems of our own bodies, guided by choices we make each day. Pharmaceuticals can manage disease and sometimes extend lifespan at the margins, yet the deepest levers for increasing health span are microbial and behavioral. Trillions of bacteria, viruses, and fungi co-evolve with us, responding continuously to diet, sleep, movement, light, and social context. When these organisms thrive, they produce hundreds of metabolites: short-chain fatty acids, neurotransmitter precursors, bile acid derivatives, that collectively reduce inflammation, bolster mitochondrial function, and tune gene expression toward repair instead of decay. Medicines usually target a solitary pathway, one molecule to inhibit an enzyme, another to block a receptor. That reductionist approach is powerful for acute conditions, but struggles to coordinate the thousands of interconnected processes that drive aging. Supporting the microbiome through nutrition, circadian alignment, exercise, and stress regulation, however, supplies a built-in polypharmacy of beneficial signals precisely adapted to our physiology. Short-chain fatty acids, bile acid derivatives, neuroactive compounds, and antioxidant enzymes act in concert, continually adjusting to our internal and external environment in a way a single pill never could. Take the elegant feedback loop between microbial metabolites and epigenetic regulation. Compounds like butyrate act as histone deacetylase inhibitors, turning on genes for cellular repair while silencing those tied to senescence. No pill replicates that nuanced gene tuning across multiple tissues. Likewise, consistent exposure to morning sunlight entering circadian clocks in both host and microbe, synchronizing metabolic and detoxification rhythms. Layered habits create synergy. Each behavior amplifies microbial output, which in turn reinforces the physiological benefits of the behavior. Behavioral interventions also compound over time. A brisk walk not only improves cardiovascular fitness, it enhances insulin sensitivity, stimulates gut motility, elevates mood modulating neurotransmitters, and encourages growth anti-inflammatory bacterial strains. And a fiber-rich, polyphenol-dense diet and those same bacteria ferment plant matter into metabolites that lower oxidative stress and support neuroplasticity. Combined with high-quality sleep, and overnight microbial cycling deepens gut barrier repair while optimizing endocrine balance. The result is a self-reinforcing loop of energy, resilience, and slowed biological aging. Personalization is another strength of the microbial behavioral model. Because every microbiome is unique, the body self-optimizes when given broad lifestyle inputs instead of fixed drug dosages. Two people may adopt identical fasting windows, yet generate distinct metabolite profiles tuned to their genetic and environmental context. This endogenous customization updates daily, something static pharmacology cannot achieve without constant adjustment. Economic and accessibility factors further highlight why behavior will drive the next wave of longevity. Fiber, sunlight, movement, cold water, and social connection are free or inexpensive, requiring no patents, complex supply chains, or prescription insurance. They scale to entire populations with minimal ecological footprint, democratizing anti-aging rather than reserving it for the affluent who can afford boutique therapeutics. Epidemiological studies confirm this communities emphasizing whole food diets, daily movement, strong social bonds, and time in nature consistently live longer and suffer fewer chronic diseases. These advantages appear before high-tech medicine enters the picture, illustrating that lifestyle patterns mediated through microbial pathways form the true first line of defense against aging. This perspective does not diminish the value of medicine. Instead, it reframes drugs as supportive tools, not primary engines of youth. Even promising compounds like senolytics, rapalogs, or NAD boosters will yield better results atop a foundation of robust microbial health cultivated through intentional routines. Our bodies evolved to interpret behavior as data, and the microbiome is the decoder translating those signals into biochemical reality, focusing first on the signals in food rhythm, movement, rest, and environment, unlocks a symphony of internal chemistry that pharmaceuticals can only aspire to imitate.